Hi class, Dr. Jim here. So you made it through the first lecture I see, and now you've conquered and gone on to the second lecture. So we're kind of jumping ahead of a couple of chapters. So chapter 2 deals with the chemistry, and we're going to talk about that in the next section of lectures. So that will be coming up in a couple of weeks. As well as chapter 3, we're kind of doing in the lab itself. So we're going to skip chapter 3 because that deals with microscopy, and we're going to be talking about that in the lab itself. And so I kind of tend to just skip that one in together so that you don't have to bother with it. But what we are going to do is kind of take a survey through the different types of microbes. And so this chapter, chapter 4, is looking at the prokaryotes. Okay, and so in this lecture, we're going to be looking at what are the prokaryotes. And so hopefully somewhere along the way you learned about what a prokaryote is. If you haven't learned what a prokaryote is, we're going to find out what one is today. Essentially, the prokaryote is a cell that is lacking a nucleus and any organelles. And so we're going to be looking in chapter 5, which is a eukaryotic cell. And again, those are cells that have nuclei and then also organelles. And so today we're sticking with the bacteria, which are the prokaryotes that don't have any, again, don't have any nucleus or organelle associated with them. We're then going to talk directly about what is made up of a bacteria cell. So when you think of a bacteria cell, it's actually a very simple cell. There's not much going on the inside. A lot of the fun stuff that we're going to be looking at is going to be inside the cell. So we're going to be talking about some of the appendages they have, they use for locomotion and genetic transfer. We'll definitely be talking about cell walls. This is going to be one of the key stains you're going to be doing all semester. And so today is really a good lecture. And if the only thing you get out of it is what is a gram positive and gram negative, I've done my job, because you guys will understand the difference between the two of them and why it's so important. We'll also look at the membranes. We'll take a few minutes and talk about membranes, look at the genomes of the bacteria, ribosomes, and then finally finish up with the shapes and arrangements of the cells. Finally, we'll finish the lecture looking at the archaea. The archaea are very similar to what bacteria are. Remember when we talked about it in chapter 1, the 3 domains. Bacteria is the first domain. The second domain is archaea. Archaea are bacteria, or prokaryotes, I should say, that live in extreme environments. So they don't really fit in the bacterial category, and they don't really fit into the eukarya category. So they're kind of in their own little place. And so we'll talk a little bit more about them. I'll show you a few pictures about these things as we go on, and kind of talk about why they are so much different than the bacteria and a eukaryotic cell. So let's begin our lecture and see where we're going and where we're headed today. So again, the thing is we need to know what is life. And so when we talk about life, we look at these things called cells, okay? And what we think of as cells is either a single or multicellular unit that's made up of cells that have common characteristics. And so we'll talk about basic shapes, and again, types of shapes of the cells. We'll talk about the internal content, the cytoplasm, and then the membrane that surrounds it, and then finally the DNA chromosome, looking at ribosomes and metabolic capabilities. When we get into the viruses, we're going to talk a little bit about what makes a virus not a live entity. And so this is going to be a really important topic. But for now, let's just focus in on the eukaryotic and prokaryotic. And really today, we're going to focus in on the prokaryotes, the ones without the nucleus. Okay, when we talk about eukaryotic cells, again, these contain membrane-bound organelles. These are the animals, plants, fungi, and protists. Again, they contain double membrane-bound nucleus with DNA. They have the organelles. And so what makes them different is that they're quite a bit larger than a bacteria cell. And when we think of prokaryotic cells, they really don't have a lot of structure on the inside. So again, these are the bacteria and archaea. They don't have a nucleus, and they're missing any membrane-bound organelles. So kind of like a mitochondria or a chloroplast or a rough ER or smooth ER. So poor little prokaryote is missing any of those things. They're kind of like what I like to refer to as a bag of water filled with some DNA on the inside. And so that's what we're going to be looking at today. All right, so some of the characteristics of life, again, having the DNA. So DNA is the structure. We're going to be talking about DNA in chapter 9, looking at the process of going through transcription, translation, and the central dogma of biology. And so that's important. We'll look at growth and development. Whether you're a bacteria cell or a human cell, we go through the process of growing up and getting bigger and larger and dividing once we've got achieved in a very large size. We'll talk about cell movement and response. Bacteria cells as well as eukaryotic cells respond to their environment, so we'll look at those things. we'll look at cell metabolism, and so we'll spend some time in Chapter 8 looking at both the process of cellular respiration and a little bit about photosynthesis, but again, the process of making ATP. We'll talk about transport again in Chapter 7 and Chapter 8. We'll be looking at transporting the molecules across the membrane. And then finally, a little bit about cell support, having these things, these internal structures that give the cell the structure and the shape. And it's important of the things that we look at in these types of things. Okay, so here's the structure of the bacteria cell. So these are the prokaryotes. And so again, most of the fun stuff on the prokaryote is on the outside. And that's going to have to do with either attachment, movement, something involved with the outside, allowing it to stick to things. Once you get inside, we'll talk more about the cell wall here in a little bit. Once you get inside, it's basically just full of cytoplasm, and inside that cytoplasm you have the DNA chromosome. Typically, bacteria only have one chromosome, unlike us where we have 46. The bacteria only has one little chromosome. Okay. We also see some inclusion bodies. We see some ribosomes and a couple other minor things in the inside, but basically that's it. It's a lot of it on the inside is the cytoplasm, the DNA, and the ribosomes, and that's pretty much what makes up the prokaryotic internal structure. We'll get to the outside first, and we'll work our way in when we describe the structure of the cell. And so these are some of the things we're going to be looking at as we go along. All right, so the first thing we're going to do is start from the outside and work our way in with the prokaryotic cells. And so the first thing we're going to look at is how do the cells move, or what are some of the things that the cells have that'll either allow them to move or attach to things. And there's really two types. The first type is used for motility, and we call this the flagella or axial filaments. And so these are periplasmic flagella that are actually inside the bacteria that help them move and kind of twitch around and move and do these different things. And we'll see this with the spirochetes especially. The other thing that these appendages are used for is to sticking to things. And so this is going to be really important. And I'm going to talk about sticking to things here, especially with your teeth here in a few minutes, and talk about some of the slimy things that you might feel on your teeth and that stuff. And that's really important for the attachment of things. And again, this can be one of those issues when you're talking about in the medical field. You use a lot of plastics, you use a lot of catheters, you use a lot of IVs and different things. And bacteria love to stick to plastics, and so that can be an issue. And so we'll talk a little bit about this and why some things stick and other things don't. And so again, two main things. One is for movement, the other is to stick to things. All right. And so flagella help the bacteria move. And so typically the way the flagella work in bacteria is that they spin like a propeller. And so you think of it, if you want to go in one direction, you spin clockwise. If you want to go in the opposite direction, you spin counterclockwise. And that's basically how the bacteria move. Now, bacteria can have flagella, either have single flagella or they can have many flagella, depending on the species of bacteria. And so, or they may not have any flagella at all. And so again, these help these guys move. And there are some bacteria that don't move at all, and so we'll look at those as well. These are some of them that invade our skin and other things like this, but we'll talk about that later on as we go through the semester. Okay, so here's a little quick YouTube video. It's just basically looking at bacteria cells under a microscope, and you're actually going to see these guys move. And so it's a minute long. Click on this after you get done watching the video, or you can pause me here and click on it and just kind of watch the video. The only reason I don't bring it in is because it makes the file way too big and it will take forever to kind of load it up and that stuff. So I leave you the link, and again, I have it as the second video in this section of Chapter 4. Okay, so make sure you watch it. It's kind of a neat little video. You'll see the bacteria just spinning around. Okay. Another thing is where, and I talked about this already, where the bacteria have their flagella. And so you have some bacteria that only have one flagella. We call this monotrichous. Trichous meaning tail-like, and so these guys have one tail. You have lophotrichous, which basically have small bunches from one site. This is typical of like a salmonella, where salmonella will have multiple flagella coming out of one end of the bacteria. You can have amphitrichous, where it comes out of both ends, and so they have flagella on both ends, as you can see here. And then you have paratrichous, which means they have flagella all over. And so again, they can move their cells in any direction depending on which flagella they move. And remember, bacteria flagella spin. They spin around as a clock. So that's kind of what we're going to think about as they move. Okay, so how do bacteria actually move? Well, again, they have a lot of taxis, so they're attracted to chemicals. So if they're attracted to food, they'll move towards it. If it's a toxin like an antibiotic, they're going to move away from it. And so they can move these ways, and we call that taxis, moving of the organism from one area to another. So if we're talking about a chemical stimuli, we're talking about chemotaxis. If we're talking about light, either moving towards the light or away from the light, that is called phototaxis. Okay, and so again, bacteria spin, and depending on which way they move, they call it either a run or a tumble, depending on what they do. So when the bacteria spin their flagella counterclockwise, so on the opposite way of the clock moving, they actually call that or term that as a run. So that's kind of the straight ahead motion, going counterclockwise. When they go clockwise in the way that the clock spins, they actually cause what is a tumble. And you can kind of see some of the patterns here where you see the bacteria kind of moving in all these different directions. And if you watch that YouTube video that I had posted, you can see the bacteria kind of spinning around and moving in different directions depending on how their flagella spin. And again, I'm not going to ask you specifically what is a run, what is a tumble. I just want you to be aware of how these guys move. And so they, depending on which direction they spin their flagella, is going to be the direction that they're going to move in. Okay. Now, some bacteria have these what are called periplasmic flagella. And periplasmic means that it's the space between the cell wall and the outside membrane. And so again, here you can see in this bacteria here, you have a cell membrane, you have a periplasmic space in the middle, and then you have a second membrane on the outside. And this is typically, or an outer sheath. And this is typically seen in a lot of gram-negatives and some other bacteria. But a lot of times we see that they have these flagella, and so the flagella is actually not outside of the organism in the sense that you see this long tail, but it's actually inside, internalized in this little sheath that they have, and it allows them to kind of flex and move around, kind of like an amoeba would, or something that would kind of crawl around. And so that's called the periplasmic flagella. And I just bring it up because sometimes you might see this in different things. A lot of times they call this motility twitching or moving of these periplasmic flagella because they kind of twitch back and forth and kind of move in kind of a movement or grouping. I don't know, where kind of blob-like movement, I guess, as you would call it. Okay. Now, one of the videos I will show is how the bacteria moves. So if you didn't watch the YouTube video, I'll play this one for you. I think this is a really good video, just showing you how the different types of bacteria move. So let's play this and see how it goes.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of 3 parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament. The basal body consists of rings that correspond to the layers of the cell envelope and therefore differ in gram-negative and gram-positive bacteria. In most gram-negative bacteria, there are 4 rings connected to a central rod. Gram-positive bacteria have only 2 basal body rings, one connected to the cell membrane and the other to the cell wall. Motility is mediated by proteins associated with the plasma membrane and basal body, and is powered by the proton motive force associated with the plasma membrane. The filament is a rigid structure which rotates to move the bacterial cell. Spirochetes are motile by means of bundles of flagella, called axial fibrils, that run along the cell within the periplasmic space. The number of fibrils varies. These axial fibrils extend from both ends of the cell within a flexible outer sheath. Spirochetes have a screw-like motion which allows them to bore into mucoid layers and soft agar media. Some bacteria lack flagella or axial fibrils, but are motile if they are in contact with a solid surface. This type of movement is referred to as gliding motility. For some gliding bacteria, secretion of a slimy polysaccharide appears to be involved. Other bacteria appear to have special proteins on their surfaces that mediate gliding motility.
So again, this video is, I just show this video because it goes into a lot more depth than I really want you to know. Like, I don't care if you know all the ring structures and everything else, but I just want to give you an idea that there are lots of different ways that bacteria can move. It's not just flagella, but they also have the periplasmic flagella in some species, and then some of them actually can glide kind of on a mucousy surface and things like this. And so there are some different methods of the way they move, but really what I want to focus in on is the flagella today. All right, so let's move on. Okay, so here's the question. Which of the following best describes the action of the prokaryotic flagellum? Does it whip back and forth? Does it extend and contract to move the cell? Does it rotate to move the cell? Does it attach to the environment and pull the cell ahead? Or is it capable of all the above? I'll give you a minute to try and answer this question. Either pause it or see if you can answer it in the time that I give you. Okay, it is actually C. If you answered C, you are correct, because basically again, the flagella spins and allows you to move either in one direction, and depending on the spin, clockwise or counterclockwise, allows the cell to move. Okay, so that's typically how we see it. We get into the eukaryotic ones, we're going to look at the whipping type of motility, like what you think about with the sperm, but that's with the eukaryotic cells. In bacteria, it rotates, it spins. Good job if you got the answer. All right, so the next structure that the bacteria can have are called fimbriae, and fimbriae look like little fine hairs. And so it kind of makes this bacteria look kind of furry under the electron microscope. And what this does is it allows it to stick. Think of it as a molecular Velcro that allows it to stick to surfaces. And you can see here E. coli cells that are stuck to the intestinal wall. We have lots and lots of E. coli that live in our intestines, and the way that they stay in our intestines is they like to stick to into our intestinal wall. And so the way that they stick is using these little fimbriae. So think of them as little molecular Velcro that they allow them to stick to the cells and the cell adhesion. Okay. The other structure that bacteria have a lot of times are pili. And pili are longer fimbriae filaments. And so they're kind of in between the size of a flagella and the size of fimbriae. Fimbriae tend to be short little hairs, where pili are kind of in between that size and a flagella. And so these are used for conjugation. Now, conjugation is kind of a funny idea. It's basically where bacteria swap DNA. And so we like to term this in the microbial world as bacterial sex, where one bacteria can swap DNA with another bacteria. Why do we even care about this? Why do we care about conjugation? Well, we're going to talk about it again in genetics, but it's an important process and where bacteria can share genetic information with one another, especially when you talk about resistance. And so if you get into the medical world, you know there's a big problem with antibiotic resistance. And one of the ways that bacteria can become resistant to things is they can swap their DNA. And so that's a real big issue. And so that's why we worry about these types of things. And one of the ways they can swap DNA is through conjugation. And so they use this to transfer. So you can see this guy has a pilus. This pilus will send the DNA over to this guy, and likewise he can send things back through this pilus. And so you can kind of see how these things will work. Okay, another thing that bacteria can have are glycocalyx, and a glycocalyx is kind of a slimy layer that they have on the outside. It's a sugar coating that they have on the outside. It can be either very rough and disorganized, like the slime layer that you see here. It's kind of rough and disorganized, and this allows for sticking things. So if you ever remember playing, maybe when you're growing up, with the thing called a wacky wall walker, it's one of those things you could throw to the wall and it kind of tumbles down. It's kind of sticky. That's kind of what a slime layer is. It's kind of a sticky, mucousy thing on the outside that allows you to stick. The other type is a more organized type structure, which we call a capsule. And these are actually very organized, and they actually are used for protection. And why would you think the bacteria needs protection? Well, once it gets inside of you, there's your immune system that's ready to eat things that are not supposed to be there. Well, a capsule prevents them from being eaten. And so that's one of the things that we see in pathogenic bacteria. A lot of times we talk about encapsulated or capsuled bacteria that actually avoid immune detection, and that's because they can't be eaten. Okay, so the slime layer is allowed to stick to things, and they make things called biofilms. And the biofilm, the best example that I can give is if you haven't brushed your teeth in a little while, you start to feel the sliminess on your teeth. Well, that sliminess is the glycocalyx that is produced by the bacteria that are stuck and growing on your teeth. And so now I know all of you are sitting there licking your teeth, thinking about the sliminess that's growing on your teeth. You might even pause it to go brush your teeth. Again, it's a natural phenomenon. It's something that happens all the time with us. And again, this is some of the things we worry about when we're talking about bacteria sticking to things, because this can cause problems, especially when we look at infections and other things like this. But this is one of those things, the glycocalyx, that allows it to stick to things. Okay. Functions, again, protect from dehydration and nutrient loss. We talked a little bit about that, inhibiting the killing of white blood cells by, again, white blood cells can't eat the ones that have the capsules. And again, the biggest thing the glycocalyx does is attachment, allows it to stick to things. And I talked to you about that, the sticking to your teeth. We call those films or mucousy films called biofilms. And again, I'm going to show you a little video here in the next minute talking about how biofilms are formed. Okay. So let's look at biofilms. So think of this as the surface of your tooth or a clean surface that maybe a plastic that's you have inserted into a patient. And this is how a biofilm will form.
Clean, abiotic surfaces immersed in liquid tend to attract and concentrate nutrients, whereas biotic surfaces such as plant or animal tissue can themselves be a source of nutrients for microorganisms. A biofilm is formed when microorganisms attach to and grow on a variety of biotic and abiotic surfaces. The initial attachment is usually reversible and often involves attachment by flagella, Fimbriae, or other cell surface structures. This is followed by growth of the microorganism and production of an exopolymer, which makes the attachment irreversible. Growth within the biofilm is not uniform. Channels develop which permit incorporation of fresh nutrients and oxygen. Metabolic activities of microorganisms in a biofilm can create changes in the microenvironment. For example, an oxygen gradient can develop that permits anaerobic metabolism near the interior and aerobic metabolism on the exterior. Nutrient and pH gradients are also produced. In the natural environment, biofilms often consist of different types of organisms that function together in the cycling of the elements. For example, breakdown of cellulose on the surface of a decaying plant may result in release of glucose and other carbohydrates, which supports the growth of non-cellulose degraders. Carbohydrates are converted to organic acids and methane by anaerobic bacteria, and the methane can then be degraded by aerobic microorganisms.
So you can see when you get a biofilm, it's not just one bacteria, but it can be multitudes of various different organisms. And that's really what's growing in your teeth. You actually have more types of bacteria in your mouth than anywhere else in your body. So you have lots of different types. And so if you suffer from bad breath or slimy teeth or whatever, that's because of the bacteria that's growing there. They're eating the sugar that you have on your teeth or from your last meal, and that's how they start to accumulate. You start to see these layers. And if you don't take care of this stuff, you don't brush your teeth, these accumulate and your body starts to form tartar, which is the stuff that you have to go in and get scraped off every time when you go to get to the dentist. And so that is these biofilms, okay? You also see this with plastics. So you'll see these things bind onto plastics. They start producing different chemicals and that attracting more bacteria, and you start to get these slimy things. So if you've worked in a hospital before, you probably know what I'm talking about, where you see sometimes where people get bacterial infections and in the plastics that they have inserted into them, either a catheter or an IV, or sometimes these things can actually close off because of the bacterial growth that you see with them. And so it's one of those issues we always deal with because we live with lots of bacteria around us. Okay, so this shows you a biofilm on a catheter, and I think, again, very important for you guys. You guys are going in the medical field, so you can kind of see the catheter surface. You can see here's some of the staph, here's some of the fungal cells, and so you get a wide distribution of different types of things that grow in them. Lots of things in the air come in, land, and start to form these biofilms, and they work together to form these films, and eventually what it can do is basically shut down the whole catheter itself. It can cause a clog, can cause infection, other things that go on. And so no matter how clean you are, it's more than likely you're going to develop a biofilm in these catheters if they're not changed on an on-time kind of basis. Okay, The next thing we're going to look at is the cell envelope, and that's basically what surrounds the bacteria. And so we're going to spend a little bit of time looking at specifically the cell wall. Now, the cell wall is very important in the bacteria. This is one of the things that is very different between bacteria and us, besides not having a nucleus in that. Because bacteria have a cell wall, our cells don't. And so a lot of the drugs that we take to kill bacteria, so the antibiotics and that stuff, target the cell walls. Because we don't have them, they're not going to be toxic to us. And so that's why we use these drugs to kill the bacteria, because they're going to be toxic to the bacteria because they have the cell walls and we don't. Okay? And so when we look at the cell envelope, basically we talk about the cell wall and the cell membrane, maintains the cell integrity. Okay? And there's two different types. We have the gram-positive and the gram-negative. And so what I want you to get out of this is, what do these cell walls look like and what's the structure? When you're talking about a gram-positive, these are going to be very thick cell walls. They're going to be very large and very brick-like pattern. Whereas a gram-negative has a very thin cell wall. And so that thin cell wall is a very weak cell wall in the sense that it's not very thick and not very structurally sound. But they also have other things that go on with it that can make it pretty nasty as well. And so we'll talk about these couple of different things as we go on with the various slides. So for the next couple of slides, we're going to be talking about the gram type bacteria, and then the gram stain, which we use to identify these bacteria. Okay, so when we look at the cell walls, again, the cell walls prevent lysis due to changing osmotic pressures. And so when we get into some of the chapters, chapter 2 and chapter 7, we're going to talk about osmosis and the movement of water. Well, one of the things that the cell wall does is prevents from the loss of water or the change of the osmotic pressure. And so these allow for bacteria to grow in lots of different environments. The cell wall is made of this protein, this sugar protein called peptidoglycan. So you can see peptido, that's protein, glycan is sugar. So it's basically a sugar protein that it's made out of. And it's a unique macromolecule that gives structure. And again, in the gram-positives, you see this repeating framework of NAM and NAG. And so you have this NAG down here, and then you have this NAM. And these structures allow for these things to be built together. And so we call these like brick-like patterns. And so in the gram-positive especially, you see lots and lots of layers of this NAM and NAG. Okay, so here's the gram-positive. Again, the size of it is anywhere from 20 to 80 nanometers thick. The thicker the cell wall, the more likely you're going to be resistant to antibiotics. So when we hear of things like MRSA, which is methicillin-resistant Staphylococcus aureus, it has a very, very thick cell wall. And that's one of the problems with it. Antibiotics cannot get in and kill the bacteria because it has such a thick cell wall. So think of that, thick cell wall. Okay, has things like teichoic acid and lipid teichoic acid, and it functions in cell wall maintenance and enlargement during cell division. And again, very thick cell walls. Some cells do have a periplasmic space. That's the space between the wall and the membrane. Most of the time, it's very, very thin, unlike the gram-negative, where you see these very large periplasmic spaces that they have. So think of gram-positive being very thick. Okay, when we look at the gram-negative, you can see here, same colors again. Here's the cell wall, very thin. We only have one layer of the NAM and NAG typically, but you have the second membrane on the outside. We call this second layer lipopolysaccharide, or LPS. Now what's interesting about this LPS is that this itself, by itself, can cause an immune response. If I injected you with purified LPS, you would feel just as bad as if you had a bacterial infection, because this stuff can trigger an immune response just as well as a bacteria can. And so it's very immunogenic, we call it. It causes an immune response, so we call it immunogenic. Okay, so the bacteria has a membrane on the outside, a cell wall, and then its normal cell membrane on the inside. And again, you can kind of see it has more periplasmic space because it has a space between the membrane and the cell wall, and then it has a second space between the outside membrane and the cell wall itself. Okay, and so again, we have a little bit more structure to the gram-negative. So think of gram-negative as a thin cell wall. Okay, all right, and you can kind of take a look side by side. Here are the brick-like patterns of the cell wall here of the gram-positive, and here's the very thin cell wall of the gram-negative, and then on top of it you have another membrane, and then you have a second membrane underneath, which is the typical cell membrane. Now both gram-positive and gram-negative both have a cell membrane, and they both sit underneath the cell wall. The difference is the gram-negatives have a second membrane on the outside of the cell wall. You don't see that with the gram-positives. Okay, so here's showing you some of the characteristics between the two. And again, I'm not going to have you sit there and memorize all these things. Really what I want you to take out of it is when you think of gram-positive, think of thick, very thick cell walls. Gram-negative, think of very thin cell walls. The other thing is that you have only one major layer. In gram-negative, you have two. And again, outside membrane you see in gram-negative, no outside membrane in the gram-positive. Both have that inside cell membrane, cell wall, and then in the gram-negatives, a cell membrane on the outside, no cell membrane in gram-positive. Okay? All right. So which of the following is not found in a gram-negative cell wall structure? Is this porins, teichoic acid, periplasmic space, lipopolysaccharides, or peptidoglycan? Give you a minute. You may have seen this when I was talking about it. You may not have spotted it. So let's see if you get it right. Okay. The answer in this case is the teichoic acids. I talked about this with the gram-positive. These are not seen in the gram-negative cell wall. Okay, so this is primarily a gram-positive type of thing. Everything else found in the gram-negative. Okay, so now we get to the gram stain, and you guys are going to become experts at doing the gram stain throughout the semester. So depending on when you watch this video, we'll be doing the gram stain either the first week or the second week of lab. I think probably the second week of lab, the way I have it scheduled for you guys. And this is going to be a very important stain. First, we're going to talk about simple stains and how they work, and the idea of it is very simple. You stain the bacteria so you can see it. The difference with the gram stain is that you can distinguish between the two types. You have the gram-positive and the gram-negative, and that's going to be the key. Okay. When you do the gram-positive, you think of thick. Now, I want you to think of a thick sponge that soaks things up. Okay. With the gram-negative, think of thin, kind of like a paper towel. It doesn't soak things up very well. Okay, or it can get overloaded very quickly. Okay, so the way that we do this is we can determine the gram-positive and the gram-negative based on what color we see with the cell walls. Okay, and so again, this is an important basis of bacterial classification, and it's a way of diagnosing and guiding drug treatment, because one way we treat gram-positive and the other way we treat for gram-negative. And this is one of the first tests that you actually will order up if you go into a nursing facility and you have a patient that has a bacterial infection. The first thing you're going to run is a gram stain to figure out is it gram-positive or gram-negative. You guys are going to be running them this year. All right. So here's the video on the gram stain technique. Again, this takes you through step by step. I'm going to be showing you this in the lab. So again, it's not that important that you watch. I think it just kind of reassures or reaffirms what you're going to be seeing in the lab. So I think it's a really good, helpful video. Again, I have it associated with it. I think it's the second or third video that you'll see beyond this one in the chapter. And again, I just highly recommend looking at it. You don't have to take notes on it. We're going to be doing it in the lab. But it's a nice way to kind of recall and see some of the steps that you're going to be doing in the lab itself. Okay. So when you do the gram stain, you actually will see two different colors. Okay, so you always start off with the crystal violet, and crystal violet is a nice deep purple stain. And so think about the type of cell wall. In gram-positive, you have a very thick cell wall. It's going to soak up the purple stain. In the gram-negative, you have a very thin cell wall. Think of the paper towel. It soaks it up, but it's not going to be, you can't hold on to it very well. It doesn't soak up as nearly as much as the gram-positive. Okay, you treat it with what is called Graham's iodine. And Graham's iodine is an iodine that allows it to really stick tightly to the cell wall. So again, with a thick cell wall, you're going to see lots of stains being stuck. Whereas in the gram-negative, you're not going to really see any more stains stuck to the cell wall. It's kind of stuck and that's it. The key step with any gram stain is the decolorizer. Okay, so when I ask this in your post-lab questions and anything else, the key step when you do the gram stain is decolorization, because that essentially removes the color from one of the bacteria. Okay, so you women out there, or maybe some of you guys that paint your nails, I don't paint my nails, but when you want to change the nail polish, what do? You dip it in a acetone or alcohol acetone solution, which removes the nail polish. And the same thing goes here. In the gram-positive, when we apply decolorizers, since you have that very spongy, like soaking up stain, the purple stays with the bacteria even though you apply decolorizer. In the gram-negative, since you have such a thin cell wall, that cell wall cannot maintain the stain. And so what happens is as soon as you apply the decolorizer, the stain goes away. What happens when most people do this, the stain, the first time and they add the decolorizer, they get really nervous because they see all their stain washing off and they go, oh no, I've ruined my stain, I got to start over. It's okay. Let me reassure you, it's going to be okay. Because again, that is supposed to happen. In some bacteria, all the stain is supposed to wash away. So don't freak out when you do this in the lab in the next couple weeks that all the stain goes away and you're going to be like, Dr. Jim, all my stain went away. What did I do wrong? And you probably didn't do anything wrong. It was due to the gram-negative nature of the bacteria. Okay, we then go in and stain with a different stain and a red stain. Now, purple is much darker than the red, and so even though you stain with the red, there's no room for the red to stick, and you don't see it because the purple just is so dark that you just don't see the red. Now, in the decolorized one, there's no stain there, so when you come back with the red stain, it's going to stain red. And so typically it doesn't stain that well, but you still see it, and it comes out kind of a reddish pink color on the bacteria. So what you associate is gram-positive, think of positive P is purple, and the gram-negative is a red or a pink color, okay? And so that's how we determine the two, by the way that this looks. Gram-positives are always going to be purple. They have thick cell walls. They soak up that purple stain. Gram-negatives, thin cell walls, they stain pink, okay, or red. And so that's how we distinguish between the two bacteria. All right. There are some bacteria out there that don't have cell walls, or they do have cell walls, but they don't stain as well with the gram stain. And so we call these the non-typical cell walls. One of the bacteria that are very important in the idea of infections and that stuff is one that causes tuberculosis. This is called Mycobacterium. And Mycobacterium has a different cell wall than the normal gram-positive, gram-negatives. They have what are called mycolic acids, and they kind of cause the gram stain not to stick very well, whether you're purple or whether you're red. And typically it does stain a little bit with the purple, but you don't get it's a very weak purple stain. So what we call these guys with the mycolic acid is an acid-fast type of stain. We actually use a different stain altogether on these guys. We actually don't do it in the lab, but if you do work with a patient that has or suspected tuberculosis infection, you're going to order up a different stain rather than doing the gram stain. You're going to do what is called an acid-fast stain, and that's primarily for these bacteria, the Mycobacterium, that have the mycolic acids. There are a few bacteria out there that don't have any cell walls together. We call these mycoplasms. They're pretty rare, but typically we see these things associated with cell culture lines and sometimes infections, these really weird infections that don't look like normal bacteria. But again, not typical, not what you're going to see most of the time when you're talking about bacterial infections, so we won't talk too much about this, but there are some that don't have cell walls together, and again, non-typical cell walls. All right. So when we look at the cell membrane, we're going to talk more about the cell membrane in Chapter 7, and so I won't spend too much time on there. But again, they have the fluid mosaic model, meaning that the membrane undulates, it moves around, it's a very fluid thing. These proteins that are stuck in the membrane can move around from side to side, move through the membrane and back and forth, and because it is a very fluid type model. And we're going to discuss more about this again in Chapter 7. Kind of remember bilipid or phospholipid, we'll talk about the macromolecules. This phospholipid membrane, very important. It has two phospholipids, and the orientation is based on the structure of the phospholipid. And again, we'll look at this in Chapter 2 and again in Chapter 7 in the next section. Okay? Once we get in the inside of the bacteria cell, we see that we have the cytoplasm. And so the cytoplasm is, again, mostly water. We also see some proteins and some other dissolved things like sugars, amino acids, and salts, but primarily it's water. And so again, what do I like to call a bacteria cell? A glorified bag full of water with some DNA and ribosomes on the inside. Okay, and so that's what you see here. Most of it is cytoplasm, and that cytoplasm is mostly water. All right, so then we look into the nucleoid. And now again, bacteria do not have a nucleus. They don't have a membrane around their DNA, but we do call it a nucleoid region where the DNA is found. Now, bacteria have one chromosome, one large chromosome, roughly about 4 to 5 million base pairs in length, compared to our chromosomes, which are about 100 million base pairs in length, so much smaller. Typically, it's a circular piece of DNA that's all tucked in as a ball of DNA. And it's kind of just floating around on the inside of that cytoplasm. So there's really no membrane keeping it in place. And again, when we get into transcription and translation, you're going to see that those things can take place simultaneously because you don't have to contend with the nuclear membrane in these cases. The other thing that bacteria have are plasmids, and plasmids are very small pieces of DNA, roughly about 10,000 base pairs at the most, sometimes only 1,000 base pairs, and typically they contain maybe 1 or 2 genes. And these genes are not important for normal metabolism, but they make the bacteria's life a lot easier. So sometimes these genes will code things that help them break down certain toxins, they can help break down certain proteins, do different things with certain carbohydrates and things like this. And again, we utilize these things to move things back and forth, and we're going to talk about that when we get into genetic engineering in chapter 10. But for now, I just want you to think about the plasmids as these extra pieces of DNA that kind of help make the bacteria's life a little easier. So take like your cell phone. Cell phone, you could live without it. I know it would be tough, but if you didn't have it, your life would still go on. I know it's shocking and everything. But if you took the plasmids away from the bacteria, again, their life would go on, but their lives would be a little bit harder, just like it would be with you. Not having the cell phone, you lose that connectivity, and so it'd make your life a little bit harder, but you would survive. Okay, same thing with these guys, and these are called plasmids. All right. The other thing that bacteria have are ribosomes, and ribosomes are the site where proteins are made. And so we're going to talk about this again. We talk about transcription and translation. Now in bacteria have 2 subunits, just like in eukaryotes, okay? But their subunits are smaller. We have a 30S subunit, which is the small subunit, and then we have the 50S subunit, okay, which is the larger subunit. These two combine, kind of you see here is the small and the large combine, and you see it forms a 70S ribosome. Now, every time I get a question, say, why is it 70S and not 80S? The reason for the S is how these guys spin down when you're talking about centrifuge. And so this S actually stands for what is called Sorval units. And Sorval units are where they line up on a gradient. And so it's not particularly a weight, and so it's not an additive where you add them together and you get the bigger weight. It's where they end up when they spin down. And so when you have a bigger unit, they spin down faster, and so they spin down lower, so they get a lower number. And again, it's where you find it on the gradient. So don't get confused and try and add these together, because what we'll see is with the eukaryotes, they have 2 subunits as well, but these are larger and larger subunits that add back together, and we'll talk about that again. So ribosomes important for proteins, 2 subunits that come together when you make the proteins. And again, we'll talk about this during transcription translation in chapter 9. Again, okay? All right. Other structures on the inside, the cytoskeleton. So bacteria have, in their cells, they have an internal skeleton like we do in our cells. We call these the actin and microfilaments. And again, they work closely with the cell wall and allows the cells to flex and contract and expand and those things, and give some kind of twitching or movement of the cells themselves. Again, I'm not going to talk much about it, but they do have some internal structures as well. Okay, dealing with that. All right, and so we have some of these other internal bacterial structures. Some bacteria can make endospores, and what I'd refer to endospores are basically these little time capsules. When the time is not right for the bacteria, they can shove their DNA into and basically survive the harsh conditions. Okay, certain bacteria can do this. Again, Clostridium, Bacillus, and the Sporosarnicea all can make spores, but very few can make spores, and these endospores are really tough. Okay, they have the vegetative cell, which is the bacteria cell. These are metabolically active and growing, and then you have the endospore. The endospore is when it's a very tough shelled organism. Okay, it doesn't really do any metabolism. It's basically a storage unit for the bacteria. So what they do is they store their DNA in for when the time is right, they'll come out again and form new bacteria cells. And I'll show you a little video of how this all works here in just a minute. But typically this is a way for them to survive harsh conditions. Okay, endospores are really, really tough structures. Okay, when we think of sporulation, we think of the hardiest of all life forms. They survive chemicals, they survive heat, they survive drying, they survive freezing. We've actually taken spores that are 10,000 years old and made new bacteria from them. It is not a means of reproduction, it's just a means for storing their DNA until the time is right to come back out again. And so think of it that way. Think of it as a time capsule. We'll store our DNA in there, and then when the time is right, we'll come back out again and make new bacteria cells. All right. And when they germinate, this is when they come back to normal cells again. So think of spores as the time capsule, and when it's ready to come out again, boom, they come out and form new bacteria cells when the time is right. All right, so this is the cycle, and I think instead of taking you through all these different cycles, I have the video on the next page, on the next slide, so I think I'll show you that, but you can kind of see how the cell goes from a live regular bacteria cell, it forms a spore, and then when the spore, the times are right again, the spore is a really tough little shell. When the time is right, the bacteria can erupt and come out and start the cycle all over again, and so you can kind of see it here, but I think you'll get more out of the video here in just a second. So let's
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell and the mature spore is released.
So again, this just kind of takes you through how the spores are made. Now this spore has the DNA on the inside, and again, that DNA is protected. This is a very tough structure. Heat, chemicals, things like this. When your Lysol says it kills 99.9% of the bacteria, this is the 0.1% that isn't killed. These spores are very, very resistant. And so these are the things we worry about. We're going to talk later on in the semester about some of the diseases that are caused by these spores, things like Clostridium, which causes gas gangrene, or tetanus, or botulism. These are all these different types of things that arise from spores getting into wounds or eating the spores and things like this. And so very tough structures. Okay, so these are the spores. We will be doing a spore stain. I'm going to try and do one in the lab for you guys so you can see it. It's kind of a tough stain to do. If we don't get a chance to do it in the lab, I will definitely show it to you in the slides just to kind of give you an idea of what these spores actually look like under the microscope. So we'll try and do that this semester as well. All right, so the last thing I want to talk about is some of the shapes and arrangements that we see with these bacteria cells. And so these are going to be important. So we need to learn the shapes and the names of the shapes, because a lot of times when we talk about the names of the bacteria, it refers to the shape that they are. So when we look at the coccyx refers to a round or spherical type bacteria. So when you see the round balls on the microscope, that is a cocci or coccyx bacteria. Okay. If the bacteria looks kind of a hot dog shape or a rod shape, we call this bacillus. This is the bacillus or rod-shaped bacteria, and E. coli is an example of this. The last type is the spirillum. These are the ones that look like spirals or corkscrews, and these are the spirilia or spiral bacteria. Okay, and again, we're going to see different diseases that are caused by each one of these different three type of bacteria, but these are the three common shapes. So make sure you know these shapes for the test: coccyx, bacillus, and spirillum. Okay, and here are the shapes. And so again, here are the round bacteria or spherical bacteria. We call these the cocci. Here are the hot dog or rod-shaped bacteria. These are called the bacillus. And then Vibrio is kind of a bent rod in this case, or kind of a comma-shaped bacteria. Here are the spirillum. They kind of have the spirals. And again, you see it. Here's a spirochete, which again, a little bit different, more spirals. And then you have some that are branching. And this gives rise to some of the molds and things like this that we're going to talk about later on. But again, we see all these different things. But the three main ones that I want you to know is coccyx, bacillus, and spirillum. Okay, so those are the three main types. All right. So we can also have arrangements of these things. And again, when we start looking at the bacteria under the microscope in the next couple of weeks, you're going to be looking at the shapes. And so you're looking at the shapes and then how are they arranged. A lot of times we'll see bacteria as singles, and so these will be single bacteria. So you can see the single bacteria just kind of sitting there. If you see two bacteria together, we call these diplo, or two bacteria, so in pairs. Okay, and then sometimes you'll see them as groups of four or boxes of four. We call these tetrads. Sometimes you'll see them like clusters that look like grapes, and so we call these clusters, or Staphylococcus, okay, or staph. And then sometimes you see long chains, so you'll see these long chains. The chains refer to strep, and so streptococci is a long chain of coccibacteria in long chains. Staphylococci are these groups of bacteria that are formed in clusters, okay, around bacteria in clusters. And again, we see this with bacilli. We see diplobacilli, we see chains of bacilli, so we call these streptobacilli. And sometimes we'll see them standing straight up like columns, and we call these palisades. And so those are the palisades bacteria as well. Okay? And I think this shows you a little quick video of the different shapes, again, what you'd see under the microscope.
Prokaryotic microorganisms exist in a variety of different cell shapes. Some are spherical, but seldom are seen as a single cell. A spherical cell is called a coccus. Pairs of spherical cells are called diplococcus, the plural being diplococci. Chains of cells are streptococci, and grape-like clusters of cells are staphylococci. Some genera, such as Streptococcus and Staphylococcus, derive their name from the most common pattern of arrangement of the cells. Many prokaryotes are rod-shaped in appearance. A rod-shaped cell is called a bacillus. The genus Escherichia consists of rod-shaped cells. The genera Bacillus and Clostridium consist of spore-forming rods. Spore formation in some bacteria causes swelling of the mother cell. There are also many other genera of rod-shaped prokaryotes. Bacilli occasionally occur as chains of cells called streptobacilli. Cells of the Corynebacterium group have a characteristic club-shaped appearance. Some bacteria have a curved rod or vibrioid appearance. An example is the genus Vibrio. Mycoplasmas are a group of bacteria that lack a cell wall. The cells of this group are pleomorphic, meaning that the cells take on a variety of different shapes. There are two groups of spiral-shaped bacteria. Spirilla are motile by means of flagella, and spirochetes are motile by means of axial fibrils located beneath a sheath that covers the entire surface of the cell. Bacteria such as the Nocardiae form branched filamentous cells. These cells reproduce by fragmentation. Actinomycetes form branched filamentous cells. This group is characterized by formation of a variety of specialized dispersal cells that form on the aerial mycelia. In some species, these cells consist of strings of individual small cells, whereas in other species, the dispersal cells are found in sacs called sporangia.
And so we'll see a lot of these things again come up when we talk about fungi and that. But for now, we'll just leave it at this. Again, there's some that are branched. But the three that I want you to know are coccus, which is a round, the bacillus, which are the rods, and the spiral, which is the spirillum. If you know those, you're good for the test. Okay, so we do classify microorganisms, and again, this is what you guys are going to be doing when we especially get to the unknown project at the end of the semester. You're going to be looking at them under the microscope, and so obviously one of the keys is microscopy or microscopic morphology. Then you're also going to look at macroscopic. We're going to talk about colony appearance. So next week when we do some of the staining, we're also going to be looking at colony formation, and again, we talked a little bit about colony formation and those things that go on and what they look like on a plate. We'll look at bacteria physiology. This is what we're going to be doing in the second half of the semester. We're going to be doing testing the physiology. How do these things grow? How do these things grow on different media? What do they look like? You can do sometimes antibody antigen tests to look at serological analysis, and then now with the genetic and molecular analysis, we can even get it down to the level of the DNA and RNA in these bacteria and figure out what they are. So lots of different ways to classify. What you guys are going to be doing in the lab are the first three. Microscopic morphology, macroscopic morphology, looking at the colors, looking at the shapes, things like this, and then finally the bacteria physiology. What do they do? Do they ferment? Do they break things down? How do they do them? What do they look like in certain plates? And so this will help you identify your unknown bacteria. All right. And again, there's a book out there called the Birdie's Manual. The Birdie's Manual is the Bible in the bacterial world. And so it basically is a dichotomous key to tell you what your bacteria is. So if you had an unknown bacteria and you knew the shape and you knew some of the characteristics, you could figure it out by going through and looking through this book. Again, it has all the bacteria that have been identified in this world, and it also has the archaea in there as well. So, and I think it's like it's a 5-volume set, so it's a very big set of books. You guys aren't going to be using it in the lab, but if there was something that was kind of a weird or wacky type of bacteria that you had no idea and you couldn't identify it, you'd go to these books and try and figure out what you had. Okay? All right, and then we can talk about species versus strain versus type, and so we're going to refer to a lot of these things. And so a species is basically the collection of bacteria. So we have lots of species. You know, this difference between Staphylococcus aureus and Escherichia coli are two different species, and so we'll talk about this. Once we get into the individual bacteria, when we talk about the species, there are actually a number of different strains of, let's say, E. coli. Okay, we have lab strains of E. coli that don't get you sick. There are other forms of E. coli that can get you really, really sick and cause you to have hemorrhaging and other things like this. We call these strains or varieties of the species because they produce different proteins. Even though they're the same bacteria, they do different things in different organisms. Okay, and then we also have some subspecies that we call types. And again, this refers to the different types of proteins they might have on the cell wall. We'll talk about this with viruses and different things like this. So lots of different things out there. What I want you to be concerned with right now is just species. Don't worry about it. I just bring this up just to show you that there are some different things, because a lot of times they'll refer to, well, this is a lab strain. What does a lab strain mean? It means it's basically been bred to be able to be safe to use in the lab. Doesn't cause disease. It's very similar to other species that are out there that are of the same species. It just does some different things in different situations. Okay? All right. And so, a round bacteria cell growing as an irregular cluster would be described as what? Streptococci, streptobacilli, staphylococci, tetrads, Yersinia, or pleomorphic. So again, what are we looking for? The first one is round bacteria cells. So what would that be? Okay, so if you looked at that, that would be one of the cocci. Okay, so it's either the streptococci or staphylococci, because that's going to be the round bacteria. The second thing we have is irregular clusters. Now, strep refers to chains of bacteria. So it wouldn't be streptococci, it would actually be staphylococci. Staphylococci refers to clusters. And so Staphylococcus aureus is a clustering. It looks like grapes. It looks like clusters of grapes on your microscope. And so you guys will see this in the next week or so when we do the gram stain. When I give you staphylococci, you're going to say, oh, I see clusters of grapes under the slide. You said exactly what I was going to see. And so you'll see that and you'll know, oh, that's like a staph, or it has the clustering type of thing. Very good for identification. All right, so the last thing, and I promise I'm done. Again, I am very wordy in that stuff and these things, and I apologize for that, but I think these things need to be just kind of hashed out so you guys understand what's going on. So the last group we're going to look at is the archaea, and these are the other prokaryotes. They are in the third domain, and the reason why they're in the third domain is because they have some characteristics of the bacteria, but they're not the same as bacteria. They have some differences, and so these are a little bit more like the eukaryotes. They have different RNA sequences and things like this. And so we just, instead of trying to make a subset of bacteria, we just decided we're going to make a third domain and call these guys archaea. And essentially, these are the weirdo bacteria that grow in very extreme environments. They have unique genetic sequences. They have unique membranes and lipids and cell walls. And so they really don't fit in the bacteria category. They don't fit in the eukaryote category. So we put them in here, the archaea category. Okay? Again, where do we see these? They live in the most extreme and hostile environments. Here you can see the red bacteria growing in the water here. These are probably salt marshes where they're actually purifying the salt out. And you can see the red archaea bacteria growing. They adapt to heat, salt, acid, pH, pressure, and atmosphere. So we'll talk a little bit about growth and growth conditions. These guys can survive very extreme conditions. If we're looking for things that grow on other planets, these are going to be the things we find because these guys can tolerate a wide range of very extreme environments, either extreme cold, extreme heat, extreme chemicals. These are what we're going to find. And I guarantee these are probably what are growing up on Mars. If we haven't discovered them yet, I'm sure we're going to discover them here in the next 5 to 10 years and say, aha, we have archaea bacteria that look very similar to the others. So maybe we'll have to come up with a new domain like the extraterrestrial archaea or something like that. But I guarantee we'll probably find cells like this on other planets. Okay? All right, so organisms of the domain archaea have peptidoglycan in their cell wall. Now, I didn't talk about the peptidoglycan, so I'll give you this one. It is actually, they do not have it, so this would be false in this case, okay? They have a different type of cell wall, and so since their cell walls are different, they're not in the bacteria category, they're in their own category called archaea. All right, so let's summarize. I told you we're done here. So the summary, prokaryotes are cells without a true nucleus or organelles, and so we call these guys the prokaryotes. The next chapter, chapter 5, we'll talk about the eukaryotes and what makes up a eukaryotic cell. The bacteria cell makeup, like I said earlier, most of the fun stuff is on the outside. The appendages, the flagella, the fimbriae, and pili are used for movement and to sticking to things. This is used for genetic transfer. The cell wall, biggest thing I want you to learn about, again, we'll do these in lab. Gram-positive, remember about the gram-positive? Thick cell walls, stain purple. Gram-negative, thin cell walls, stain red. The genome of the bacteria cell is one circular piece of DNA. And again, they do have plasmids. Again, the plasmids are not vital for life. They're kind of like your cell phone. You could live without them. Bacteria could live without these. They just make their lives easier. Okay. Ribosomes inside the bacteria cells are smaller. They're 70S compared to the eukaryotic ones, which are going to be 80S, which are bigger. Okay. Smaller subunits. And so, again, we'll talk about targets and things like this with antibiotics. And then we talked about the shapes. Remember the shapes. Cocci are the round. Bacillus are the rod, and spiral are or spirillum is the spiral. Okay, remember those three things. Arrangements, again, the biggest arrangements are the chains, which is strep, and then the clusters, which is staph. And then you can also think of the diplo, which is 2, the tetrads, which are 4, things like this. Okay, but the two main ones, strep and staph. If you remember that, you're good. Okay, and then the last thing is the archaea. The archaea are the extreme bacteria. They're in their own environment, or they're in their own domain type of thing. They're in hostile environments. They have different cell walls, cell membranes than the normal bacteria, which make them different, and that's why they're in this category. Okay, we've come to the end of the video. If you have any questions, feel free to ask them in our lab section. Otherwise, you can email me at any time, and I'll be happy to ask these or answer your questions. Okay, so with that, I'll see you next time. Thanks for watching.